Mathematics 1, Mathematics 2. Let's see first objective biology 1. In this subject, all topics are given. Also we cover whole textbook and biology 1 subject. Now for example, we see the topic is cell division. In this topic, two parts are given. First is introduction and second is summary. Now here see the first point is introduction. In introduction, we not only give an introduction but also we include text, voice, images, videos and animation, which will help to understand topic very easily.
Lesson 4 Cell Division Introduction. Cell reproduction is an example of pervading, ubiquitous role that the genetic system, DNA, plays in all life processes. The genes and their regulatory mechanisms determine the growth characteristics of the cell, and also when or whether these cells divide to form new cells. Every cell arises from the pre-existing cell according to the cell theory. A single cell zygote divides for so many times to form the somatic, that is, body structure of an organism. All living organisms can grow and develop because of cell division. Let us learn about how the cells divide, when does it divide, etc. Things we will learn. Cell cycle. Two main phases of cell cycle, interphase and mitotic or meiotic phase. Types of cell division in eukaryotes, mitosis, mitosis and meiosis. Elaboration. Cell cycle. A cell grows to a certain maximum size. Then it divides to form 2 or 4 daughter cells. The daughter cells formed after the division of a mother cell grow in size and divide again. This continuous process is called as the cell cycle. The generation time is the period between two successive divisions. A series of changes occur in the life of a dividing cell. These sequential events constitute the cell cycle. The period from the beginning of one cell division to the beginning of next cell division forms the cell cycle. The cell cycle consists of two main phases, interphase and or mitotic or meiotic phase. Interphase. This was formerly called as resting phase, but it is now called as preparatory phase. Because the cell is highly active during this phase and many metabolic changes occur in the cell during this phase. The interphase can be further divided into G1 phase, S phase and G2 phase. G1 phase. First growth phase, also called the post-mitotic gap phase. As we have stated in the introductory part, reproduction begins in the nucleus itself, and it is the genetic material that decides the fate of the cell. Mitosis can take place only after the replication, that is duplication of DNA has occurred. The G1 phase involves synthesis of enzymes and energy molecules required for DNA replication. The DNA begins to be duplicated some 5 to 10 hours before mitosis. The two exact replicates of DNA are produced. These replicates in turn become the DNA in the two new daughter cells that will be formed at mitosis. The principal enzymes for replicating DNA are a complex of several enzymes called DNA polymerase and DNA ligase, which is responsible for forming double-stranded helical structure. During G1 phase, proliferation of cell organelles takes place, or cell may withdraw from cell cycle and enter in the GO or quiescent phase. The cell in GO may re-enter the G1 phase or may get differentiated and become permanent cell, that is non-dividing cell. At GO phase, if mutation or abnormal activation of cellular genes that control cell growth and cell mitosis occurs, there are chances of developing cancer or tumor in which there is uncontrolled growth of abnormal cells. The abnormal genes are called oncogenes. S phase, synthetic phase or phase of synthesis. Duplication or replication of DNA. Nucleus becomes double in size. Histone proteins required for organization of chromosomes are also produced. G2 phase, second growth phase, also called pre-mitotic gap phase or growth phase. It involves the synthesis of the tubulin, the spindle protein and RNA. Duplication of cell organelles like mitochondria, chloroplasts, etc. The centrioles duplicate to form two centrosomes. Now, the cell is ready for mitosis. Types of cell division. The cell division in prokaryotes is atypical, that is binary fission. In eukaryotes, the cell division is typical and may be of the types amitosis, mitosis and meiosis. Amitosis. Here, the nuclear membrane is retained throughout the division. The nucleus elongates, enlarges and divides directly. So, it does not ensure equal distribution of genetic material in the daughter cell. It may lead to structural and functional irregularities. Meganucleus of paramecium, fetal cells, endosperm cells, etc. show this type of division. The division of nucleus is followed by division of cytoplasm, that is cytokinesis, and two daughter cells are formed. Mitosis. The actual process by which the cell splits into two new daughter cells is called mitosis. It ensures equal distribution of genetic material among the daughter cells. Mitosis is required for growth and wear and tear recovery. As this division results in the formation of somatic cells, it is also called as somatic cell division. Once each chromosome has been replicated to form two chromatids, mitosis follows automatically within an hour or two. Mitosis maintains chromosome number in daughter cells equal to that of mother cell. Hence, it is also called as equational division. Two pairs of centrioles lie close to each other near one pole of the nucleus. See the diagram. These centrioles, like the DNA and chromosomes, had also been replicated during interphase shortly before the DNA replication. Each centriole is a small cylindrical body about 0.4 micrometer long and about 0.15 micrometer in diameter, consisting mainly of 9 parallel tubular structures arranged in the form of a cylinder. The two centrioles of each pair lie at right angles to each other. In animal cells, there are distinct centrosomes and the kinetic spindle is organized with reference to the centriole. Such division is called as centric. It is acentric in plant cells. Shortly before mitosis is to take place, the two pairs of centrioles begin to move apart from each other. This is caused by successive polymerization of protein microtubules growing between the receptive centriole pairs and actually pushing them apart. The complex of microtubules connecting the two centriole pairs is called a spindle. The term mitosis was coined by Walther Fleming, a German biologist. The events involved in the cell division are divided into two stages: karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. In karyokinesis, the nucleus undergoes a series of changes to form two daughter nuclei. Steps in karyokinesis: prophase, metaphase, anaphase, telophase. Prophase. Following changes occur during prophase. Chromatin network condenses and the chromosomes which in interphase consist of loosely coiled, thin, long strands, single-stranded, monad chromosomes. become condensed into well-defined chromosomes. Each of them shows identical chromatids held together by a centromere, dyad chromosome. Throughout prophase, chromosomes undergo dehydration and cooling to become thick and short. Nuclear membrane and nucleolus begin to disappear. In animal cells, the daughter centrioles start moving away from each other till they occupy the polar position. Prophase is the longest phase. Metaphase. The condensation of chromosomes is completed. The chromosomes, which have become thick and short, get organized along equatorial plane of the cell. Thus, this stage is called as metaphase. Meta is middle and phase is state. Formation of kinetic spindle occurs. The spindle is made up of contractile proteinaceous chromosomal fibers and interpolar fibers. It is called mitotic apparatus. In animal cells, the spindle fibers grow radically away from each of the centriole pairs, forming a spiny star called the aster in each end of the cell. Some of the spines penetrate the nucleus and play a role in separating the two sets of chromatids during mitosis. The radiating spindle fibers are called as astral rays, and thus in animal cells, the mitosis is called as astral mitosis. In plant cells, the asters are absent and hence it is called as anastral mitosis. Anaphase. Shortest phase of karyokinesis. Centromeres divide into two and the chromatids get separated. Each separated chromatid is now called as daughter chromosome. The kinetic spindle now has four types of fibers made up of microtubules. They are astral rays radiating away from the centriolar pairs towards the periphery of the cell. Interpolar fibers extending between the opposite pairs of centrioles. Chromosomal fibers connective centromeres to respective poles. Interchromosomal fibers or interzonal fibers appear between the centromeres of daughter chromatids. The chromosomal spindle fibers undergo contraction and pull the daughter chromosomes to the respective poles, while interchromosomal fibers elongate, pushing the daughter chromosomes till they reach the respective poles. The chromosomal fibers contain a protein called actin in addition to tubulin. Actin is one of the contractile proteins of muscle. It is presumed that the chromosomal fibers might interact in a sliding way with the astral rays, causing the pulling force. Telophase. The two sets of daughter chromosomes are now pulled completely apart. Daughter chromosomes uncoil and undergo hydration to form chromatin network. The mitotic apparatus dissolutes and a new nuclear membrane develops around each set of chromosomes. This membrane being formed from the portions of endoplasmic reticulum that are already present in the cytoplasm. Nucleolus reappears in each newly formed nucleus. Cytokinesis. Division of cytoplasm of the mother cell. A cell pinches in two, or there is furrowing of plasma membrane in animal cells. This is caused by a contractile ring of microfilaments composed of actin and myosin, the two contractile proteins of the muscle. The furrow deepens and the daughter cells are formed. This process is called cleavage. In plant cells, it takes place by formation of cell plate which extends from center to the periphery. Importance of mitosis. As mitosis leads to the formation of somatic cells, it is very important for growth, development, healing, and repair of multicellular organisms. It maintains chromosome member of parent cell in daughter cell. Mitosis brings about equal distribution of genetic material and maintains characteristic of species. In asexual reproduction of unicellular organisms, it avoids functional irregularities by equal distribution of genetic material. Now, let us talk about the third type of a typical cell division, that is meiosis. Meiosis is termed as reduction division, which involves halving of the chromosome number. In this process, the chromosomes duplicate only once, but the cell division takes place 2 times. It forms 4 haploid cells. Meiosis occurs only in special diploid cells. It leads to rejuvenation and recombination. It occurs in the formation of spores, fungi, and cryptogams. The meiotic division is responsible for evolution and diversity. It occurs during gamete formation in animals. The meiotic division involves 2 successive divisions, Meiosis I, reduction division, Meiosis II, equational division. Both divisions involve karyokinesis and cytokinesis. Meiosis I, reduction division. Interphase, preparatory phase similar to that in mitosis, involves DNA replication. Karyokinesis, prophase I, most complicated and the longest phase involving 5 substages, viz. leptotin, zygotin, pakitin, diplotin, and dikinesis. Leptotin, also called as leptonema. Leptos means thin and nema means thread. The chromatin network condenses and resolves into long and thin thread-like chromosomes. Each chromosome is with 2 chromatids, but it appears as a single thread due to condensation. Zygotin or zygonema. Zygo means pair. The homologous chromosomes begin to pair lengthwise. The morphologically and genetically similar chromosomes, one paternal and other maternal, present in the diploid cell are called homologous chromosomes or homologs. This pairing of chromosomes is called synapsis. The pairs are called bivalents. Pakitin or pakinema. Pacus means thick. As the condensation of chromosomes progresses, they become short and thick. Each of the homologous chromosomes shows 2 chromatids, so that the bivalents appear to be composed of 4 chromatids, that is tetrad. The twisting of homologs results in breakages and rejoining of the chromatids. Crossing over, that is exchange of genetic material between non-sister chromatids of homologous chromosomes, takes place. Crossing over results in genetic recombination of parental characters, thus resulting into variation. Chiasmata are the points where crossing over takes place. Diplotin. Diplos means 2, nema means thread. Homologs start repelling each other and begin to separate. They remain attached at the chiasmata, that is crossing over point. Nucleolus and nuclear membrane start disappearing. Dikinesis, condensation and shortening of chromosomes continues. The separation of homologs proceeds and the chiasmata gets shifted to the end of chromatids. This process is called as terminalization. The nucleolus and nuclear membrane completely disappear. Kinetic spindle becomes apparent. Metaphase 1. Spindle formation is complete. Bivalents move and arrange themselves at the equatorial plane. Chromosomal fibers extend from the poles to the centromeres of corresponding homologs. Anaphase 1. The chromosomal fibers contract and pull the homolog. Interchromosomal fibers appear and extend. Because of this, homologous chromosomes start moving towards opposite poles. Centromeres do not divide. Disjunction of homologous chromosomes occurs, that is, homologs which are still attached at chiasmata finally get separated. At the end of anaphase 1, half the number of chromosomes gather at respective poles. Telophase 1, uncoiling of chromosomes to form chromatin. The nuclear membrane and nucleolus reappear forming two daughter nuclei. Cytokinesis 1, division of cytoplasm. The plasma membrane constricts in the middle in animal cell, while in plant cell, a cell plate is formed to form two daughter cells. Each daughter cell has single set of chromosomes, that is, haploid. Now, let us know about the equational division, that is meiosis II. It starts after a very short period after meiosis I. This resting period between meiosis I and II is called interkinesis. In some cases, this period may be absent. The karyokinesis. It involves prophase II, metaphase II, anaphase II, and telophase II. The haploid daughter cells formed at the end of meiosis I undergo meiosis II simultaneously. Prophase II. Chromosomes with chromatids become distinct. The nuclear membrane and nucleolus disappear. Metaphase II. Spindle formation. Movement of chromosomes towards equatorial plane. Chromosomes get connected to the respective poles by chromosomal fibers. Anaphase II. Division of centromere of each chromosome. Separation of chromatids to form daughter chromosomes. Interchromosomal fibers are formed between the centromeres of daughter chromosomes. The daughter chromosomes move towards opposite poles due to contraction or shortening of the chromosomal fibers and elongation of interchromosomal fibers. Telophase II. The daughter chromosomes at the poles start uncoiling. The nucleolus reappears. The nuclear membrane is formed around each group of chromosomes, forming two daughter nuclei. Cytokinesis II. Following telophase II, the division of cytoplasm of each cell forms two daughter cells. Thus, at the end of meiosis II, four haploid daughter cells are formed. Significance of meiosis. As meiosis produces haploid gametes for sexual reproduction, it maintains the chromosome number constant for a particular species. When two haploid gametes fuse, a diploid zygote is formed, thus maintaining the chromosome number. Meiosis accounts for variations and evolution as it involves genetic recombination due to crossing over or exchange of genetic material between non-sister chromatids of homologous chromosomes. Thus, meiosis plays an important role in creating biodiversity.
We are now to the evolution and after evolution, we are see the summary. In summary, you can do more study in short period.
Summary. Life is maintained because every new cell arises from the pre-existing cell. This is achieved through the process of cell division. Cell division is a process by which a cell divides into two or four daughter cells. A series of events occur in sequence in the life of a dividing cell. It is called cell cycle. Replication of entire genetic material, that is DNA, takes place before the cell divides. The cell cycle includes a long interphase, that is preparatory phase, and a relatively shorter M phase, that is mitotic or meiotic phase. Interphase is divided into G1 phase, S phase, and G2 phase. In G1 phase, synthesis of enzymes take place. In S phase, DNA replication takes place. And in G2 phase, synthesis of tubulin RNA duplication of cell organelles take place. Cell division in eukaryotes. Amitosis, mitosis and meiosis. A mitosis is seen in paramecium, endosperm cells, fetal cells. Nuclear membrane retained throughout the cell division. Mitosis and meiosis, two stages, karyokinesis and cytokinesis. Mitosis is called somatic cell division, and also it is equational division as it maintains chromosome number in daughter cells equal to that of mother cells. The first stage of mitosis is karyokinesis or nuclear division, which is further distinguished into prophase, metaphase, anaphase and telophase. In animal cells, cytokinesis occurs by furrowing of plasma membrane, that is cleavage. In plant cells, it takes place by formation and extension of cell plates. Mitosis plays an important role in growth, development, healing and repair of multicellular organisms by forming new somatic cells. Mitosis plays a significant role in asexual reproduction by unicellular organisms. It ensures equal distribution of genetic material and maintains characteristics of species. In meiosis or reduction division, chromosomes duplicate only once, but the cell divides twice, resulting in the formation of 4 haploid cells. Meiosis is a complex division. Two successive divisions of meiosis are Meiosis I or reduction division, Meiosis II or equational division. Under meiosis I, karyokinesis includes prophase I, leptotin, zygotin, packaging, diplotin and dikinesis, separation of homologues or terminalization. Metaphase I, anaphase I, telophase I. Prophase I is highly complex. Under meiosis II, simple division, similar to that of mitosis, involves karyokinesis and cytokinesis. Meiosis produces haploid gametes, so it is a significant process for sexual reproduction. It helps to maintain chromosome number constant for given species, despite fusion of gametes. Packaging stage involves crossing over, that is, exchange of genetic material between non-sister chromatids of homologous chromosomes. So, meiosis is significant as it allows genetic recombination of parental characters, leading to variations. It makes the way for evolution and diversity.
Now friends, the biology first, this chapter is closed. After that, we will go into next subject, is biology second. The biology the biology second, in this subject, study is given. In study, all topics are given. For example, we see the topic of structure of atom. Let's see the topic, human respiration. In this topic, introduction and summary is given. First, we see the introduction of human respiration. In introduction, we see that we not only given technical information but also we add background, voice, images, videos, and animation, which will create your interest to build more skills and improve your knowledge.
Human respiration. Introduction. You have learned that every cell in our body requires oxygen, which oxidizes the organic compounds, which are obtained from the food we eat, to liberate chemical energy. Oxygen is very essential for survival, as we all know. If the vital organs do not get oxygen for a longer time, it is life-threatening. In this lesson, let us learn about the structure of human respiratory system, the physiology of respiration, chemical reactions involved in the respiration process, disorders of respiratory system, etc. Things we will learn. What is respiration? Organs of respiratory system, mechanism of respiration, breathing, inspiration and expiration, events occurring during transport of gases, respiratory disorders. Elaboration. What is respiration? Respiration is an energy liberating process. It is chemically a slow combustion process in which the organic compounds are burnt, that is, oxidized, and energy is released. Here, burning should not be considered as what it literally means. It is not that the organic compounds are set to burn at once by combining with oxygen, which might result into light emission and heat. If it happens so, it will lead to cell death. Respiration is a stepwise process. It involves stepwise breaking of the molecular bonds of the compounds, and so many complex chemical reactions occur in our body in coordination with each other. So we can define respiration as a biochemical process by which organic compounds, that is foods, are oxidized to liberate chemical energy. The energy liberated is trapped in chemical bonds of adenosine triphosphate, that is ATP molecule. Thus, ATP is a high energy compound and biologically useful energy can be obtained from it. This energy is required for all the life processes. When a cell requires energy, ATP gets hydrolyzed to form ADP, that is adenosine diphosphate, and energy is released. C6H12O6 plus 6O2 gives us 6CO2 plus 6H2O plus 38 ATP. Respiratory organs supply oxygen and remove carbon dioxide and water. The transport of gases takes place through blood. Organs of respiratory system. They include nostrils, nasal chambers, pharynx, larynx, trachea, bronchi, bronchioles, lungs, diaphragm, and intercostal muscles. Nostrils and nasal chambers. Nostrils are the external openings of the nose through which oxygen, that is air, is taken inside the body, and carbon dioxide rich air and water vapor are expelled out of the body. They are also called external nares. A pair of internal openings into the pharynx are called internal nares. Internally, they are lined by mucous membrane. Nasal chamber is the space between the external and internal nares, and it shows presence of ciliated epithelium. Cilia drive out any foreign particles that can enter along with air. Nasal chamber is divided into right and left parts by a cartilage called as mesenteroid. Nasal chamber is divided into three regions: vestibule, anterior-most part with hair to trap dust particles; respiratory part, highly vascularized, it warms and moistens the air; sensory part, lined with sensory epithelium for small detection; pharynx, vertical tube about 12 cm in length in which the respiratory and food passages cross each other by two separate passages. The upper part of pharynx is called nasopharynx, which conducts air, and lower part is called laryngopharynx or oropharynx, conducting food to esophagus. Tonsils made up of lymphatic tissues are present in pharynx that kill the bacteria trapped in mucus. Larynx, sound producing organ. In males it is called as Adam's apple as it increases at the time of puberty and can be noticed in the neck region. From the pharynx, air enters the larynx through an opening called glottis, guarded by a flap called epiglottis. It prevents the entry of food into the respiratory passage. Two folds of elastic tissue along the sides of the glottis are called vocal cords for sound production. Trachea, that is windpipe, 12 cm long, 2.5 cm wide, lies in front of the esophagus and extends downward into the neck. The wall of trachea is made up of fibrous muscular tissue supported by C-shaped cartilage rings, 16 to 20 in number, which make the trachea rigid. Internally, it is lined with ciliated epithelium and mucous glands. Foreign particles are immediately expelled out by coughing action. Dust particles trapped in mucus are swept towards larynx by ciliary action. Bronchi and bronchioles. The distal end of trachea is divided into two bronchi behind the sternum. Each bronchus is supported by a complete ring of cartilage and it enters into the lung of its respective side. On entering the lung, each bronchus further divides into secondary and tertiary bronchi, and tertiary bronchi divides into many minute bronchioles, which are without cartilage rings. Each bronchiole ends into a balloon-like alveolus, grape-like sacs, which make the lungs spongy and elastic. Lungs, paired, hollow, elastic, located in thoracic cavity, and each one enclosed in pleural sac. Important respiratory organ. A lung is made up of two membranes, outer parietal and inner visceral. The cavity enclosed is called pleural cavity, filled with pleural fluid, which lubricates the pleura and prevents their friction. Lungs are pink, elastic, distensible, highly vascular. Left lung is bilobed, while the right is trilobed. In the interior, there are many lobules which contain the alveolar ducts, alveolar sacs, spherical thin-walled sacs, each with about 20 alveoli looking like grapes, and alveoli. Alveolar wall is very thin, 0.0001 millimeter, and made up of simple, non-ciliated squamous epithelium. It has collagen and elastin fibers, which makes the alveoli very flexible. Alveoli are supplied with the network of pulmonary capillary. Alveoli have elastic walls and their diameter is about 0.1 millimeter. About 750 million alveoli are present in human lung, and they increase the surface area about 50 times more than the surface area of skin for gaseous exchange. Mechanism of respiration. Respiration includes taking in oxygen and giving out carbon dioxide. Air is taken in and given out by the breathing process. Breathing includes inspiration and expiration. Inspiration, active process, intercostal muscles contract and pull the ribs outward, increasing the space in the thoracic cavity. Sternum is also raised and diaphragm contracts and becomes almost flat, further increasing the space in the thoracic cavity. Lungs expand, their volume increases and pressure on them decreases. It makes the atmospheric air to rush into the lungs through the respiratory tract to make the pressure equal. Thus, air enters the lungs in inspiration. Expiration, passive process, intercostal muscles relax, pulling the ribs inwards. The diaphragm relaxes, regaining its dome shape. This reduces the volume of thoracic cavity. The lungs get compressed and air pressure inside them increases, causing air to rush out through the nasal chamber and nostrils to make the inside and outside pressure equal. Alternate inspiration and expiration, that is breathing cycle, speeds up the rate of gaseous exchange. Medulla oblongata controls breathing. The breathing cycle or respiratory cycle occurs 16 to 20 times per minute. Events occurring during transport of gases. Respiratory gases get transported, that is oxygen and carbon dioxide, in the following events. External respiration. It includes the respiratory processes which occur in lungs. Oxygen from the lungs diffuses into the capillaries and carbon dioxide from the capillaries diffuses into lungs. External respiration has three events: exchange of gases, formation of oxy-haemoglobin, release of carbon dioxide. Exchange of gases. Concentration of oxygen is higher in the inspired air than in the alveolar blood, and concentration of carbon dioxide is higher in the alveolar blood than in the inspired air. This causes exchange of oxygen from the air into the blood and carbon dioxide from alveolar blood into the lung air, which is then exhaled out. Formation of oxy-haemoglobin. Hemoglobin is a respiratory protein pigment. Oxygen combines with it and oxy-haemoglobin is formed, unstable, Hb plus 4O2, Hb4O2. Hemoglobin plus oxygen, oxy-haemoglobin. Release of carbon dioxide. Carbon dioxide is brought by the blood from the tissue cells in the form of sodium and potassium bicarbonates in the blood plasma. Some amount of carbon dioxide is also brought by hemoglobin in the form of carbaminohemoglobin. Breakdown of carbonates into carbonic acid is formed. NaHCO3, that is sodium bicarbonate, in presence of hydrogen plus gives Na plus, that is sodium ion, plus H2CO3, that is carbonic acid. KHCO3, that is potassium bicarbonate, in presence of hydrogen ion gives potassium ion, that is K plus carbonic acid, that is H2CO3. H2CO3, that is carbaminohemoglobin, gives H2O plus carbon dioxide. Also releases CO2. HbCO2 gives us Hb plus CO2. CO2, that is carbon dioxide formed, is then released into the air. Internal respiration. Respiratory process that takes place in tissue cells. Oxygen brought by blood is given to the tissue cells and carbon dioxide from tissues is passed to blood. The unstable oxy-haemoglobin after reaching the tissue cells breaks down to form oxygen and hemoglobin. Carbon dioxide, which is passed into the blood, dissolves in water to form carbonic acid, which reacts with sodium and potassium to form NaHCO3, that is sodium bicarbonate, and KHCO3, that is potassium bicarbonate, respectively. CO2 plus H2O gives H2CO3, that is carbonic acid. About 80 to 85% carbon dioxide is carried by the blood in the form of bicarbonates. A small amount of carbon dioxide combines with hemoglobin to form carbaminohemoglobin. Some carbon dioxide dissolves in plasma and carried to the lungs. Cellular respiration. As you know, respiration is the energy releasing process. It involves oxidation of food inside the cells, resulting in ATP production. This energy stored in the form of ATP is utilized for all the vital processes. ATP production occurs mainly into mitochondria. CO2 and H2O, that is water vapor, are transported to the lungs by blood. CO2 and water vapor are the byproducts formed in the respiratory process. Some amount of heat is also released during this process, which is useful in maintaining the body temperature. Asthma, inflammatory disease caused due to overreactivity of the airway smooth muscle. The muscle layers of bronchi and mucous membrane become thickened, and enlargement of the mucous glands causes narrowing of the airway, reducing the airflow in the lower respiratory tract. Bronchospasm, that is spasmodic contraction of bronchial muscle, constricts the airway, and excessive secretion of thick, sticky mucus thereby further narrows the airway. Lungs become hyperinflated due to impaired expiration, leading to dyspnea, difficulty in breathing, and wheezing. Duration of asthmatic attack varies from few minutes to hours. Status asthmaticus, and in severe attacks, mucus plugs may obstruct the bronchi, leading to hypoxia, that is low oxygen levels, acute respiratory failure, and may prove fatal. Asthma may get precipitated by non-specific factors like cold air, smoking, pollution, upper respiratory tract infection, emotional stress, physical stress, etc. Pharyngitis. It is the inflammation of the upper respiratory tract. It can be viral or bacterial. Streptococcus pyogenes, that is cocci, are the common cause of inflammation of palliative tonsils, that is tonsillitis, and walls of pharynx, that is pharyngitis, generally seen during changing seasons. Laryngitis. Inflammation of larynx due to viral or bacterial infection, common in children. Symptoms: husky voice initially, aphasia in severe infection, rawness of throat, stridor in children, irritating cough, etc. Cause: vocal misuse or overuse causes acute laryngitis. Allergic rhinitis, that is hay fever. In this case, atopic hypersensitivity, that is immediate hypersensitivity, develops to foreign proteins, that is antigens. Example: pollen grains, feathers, animal dander, mites, etc. This causes acute inflammation of nasal mucosa and conjunction, and due to this, rhinorrhea develops. Excessive watery exudate from the nose. Many people have allergies to certain foodstuffs, oils, or certain fragrances. There may develop redness of the eyes and excessive secretion of tears in case of allergic rhinitis. This allergic rhinitis tends to run in families, but the genetic factor is not yet identified. Other forms of atopic hypersensitivity are childhood onset asthma, eczema in infants and young children, food allergies. Many people develop skin rash due to allergy to certain foodstuffs. Pneumonia. This means infection of alveoli. When the microbes succeed in crossing the barrier of all the defense mechanisms of the body, like mucous membranes, inflammation which makes the infection localized and hence restricted from further proliferation or humoral response, that is antibody production, etc. Then the inhaled or blood-borne organisms can reach and colonize the lungs and grow on account of the host tissue. Some of the causes of pneumonia are as follows. Impaired coughing. It is an effective cleaning mechanism. It may get impaired by unconsciousness, damage to respiratory muscles or nerves supplying the respiratory muscles, or painful coughing. The accumulation of respiratory secretions may harbor germs as they are well nourished in this medium. Tobacco smoking impairs ciliary action and can depress macrophage activity, which are the soldier cells. Inhaling noxious gases, infection also cause impairment of ciliary action. Macrophage activity is also depressed by anoxia, alcohol, and oxygen toxicity. Other factors, lowered resistance of the host may prove a chance to normal respiratory flora to become opportunistic pathogen. Leukopenia, that is less number of leukocytes which are fighting cells. Chronic diseases like cardiac failure, cancer, chronic renal failure, Streptococcus pneumonia, particularly causes lobar pneumonia, Klebsiella pneumonia, Rod's gram-negative bacteria. Mycoplasma, very common, affects mainly children and young adults. Some viruses and protozoa also cause pneumonia. In case of HIV-infected patients, Pneumocystis canini infection to lungs is very common due to hampered immune system. Immunosuppressant drugs are also one of the causes of pneumonia. Occupational lung diseases. These diseases are caused by inhaling small particles of atmospheric pollutants at workplace. These small particles are carried in inspired air to the level of the respiratory bronchioles and alveoli, where they can only be cleared by phagocytosis. Larger particles are trapped by mucus in the upper part of the respiratory tract and expelled by ciliary action and coughing. Common occupational lung diseases include silicosis, asbestosis. Silicosis is caused by long-term exposure to dust containing silicon compounds. High-risk industries are granite, slate, sandstone, mining, quarrying, glass and pottery work, sandblasting, stone masonry. The silica particles accumulate in the alveoli. These particles are ingested by macrophages and are actively toxic to these cells. The inflammatory reaction is triggered when macrophages destroy the particles. This results into significant fibrosis. Silicosis is the predisposing factor to the development of tuberculosis, which can lead to tubercular bronchopneumonia and military TB. Lung tissue gets destructed gradually, and this leads to progressive reduction in pulmonary function, pulmonary hypertension, and heart failure. Asbestosis. It develops usually after 10 to 20 years of exposure to asbestos fiber. May develop after only 2 years in some cases. Blue asbestosis is very dangerous.
Now, we have figure next part is summary. This summary will be important concept in the respiration. By using summary, we can do more studying software.
Summary. Respiration can be defined as a biochemical process by which organic compounds, that is foods, are oxidized to liberate chemical energy. Carbon dioxide is expelled out and energy released during oxidation is stored in the form of ATP molecules. Human respiratory system includes nostrils, nasal chamber, pharynx, trachea, bronchi, bronchioles, lungs, diaphragm, and intercostal muscles. Nostrils or external nares are the external openings of the nose through which oxygen-rich air is taken inside the body and carbon dioxide-rich air containing water vapor is expelled outside the body. The space between external and internal nostrils is known as nasal chamber, divided into right and left parts. Pharynx is a short vertical tube in which the respiratory and the food passages cross each other. Larynx is a sound-producing organ. Air enters the larynx through an opening called glottis, which is guarded by the epiglottis. It prevents the entry of food particles into the respiratory passage. Along the sides of glottis are two folds of elastic tissue called vocal cords which produce sound. Trachea or windpipe lies in front of the esophagus. It is internally lined by ciliated epithelium and mucous glands. It is supported by C-shaped cartilage rings. It passes air into the bronchi. Trachea divides into two bronchi and each bronchus enters the lung of its respective side. Bronchi are supported by complete rings of cartilage. Bronchi divide into bronchioles without cartilage, each ending into alveolus. Lungs are paired, hollow, elastic organs located in the thoracic cavity. Left lung has two lobes and the right lung has three lobes. Each lobe consists of bronchioles which terminate in a bunch of spherical thin-walled alveolar sacs, each with about 20 alveoli. Alveoli are supplied with the network of capillaries and they increase the surface area for exchange of gases. Mechanism of respiration includes the steps like breathing, external respiration, internal respiration, and cellular respiration. External respiration takes place in lungs, internal respiration takes place in tissue cells, and cellular respiration is ATP formation. Respiratory disorders include asthma, pharyngitis, laryngitis, allergic rhinitis, that is hay fever, pneumonia, and occupational lung diseases like silicosis and asbestosis. Asthma is an inflammatory disease of the airways associated with episodes of reversible overreactivity of the airway smooth muscle. Bronchospasm constricts the airway. Allergic rhinitis is caused due to atopic hypersensitivity to foreign particles, causes rhinorrhea. Pneumonia is the infection of the alveoli. It is caused due to the impaired cupping and damage to the epithelial lining of the tract. Occupational lung diseases are caused by inhalation of atmospheric pollutants at workplace. Example, silicosis, asbestosis. In silicosis, the inflammatory reaction is triggered when the macrophages destroy the silica particles. This results in significant fibrosis.
One of the subjects is given. After that, we will go to the subject and the new subject is in conventional study history of in study large of the structure. For example, in this topic is considered as pattern. In this topic is covered by the given. In introduction, the term introduction is given. On given. Introduction is not only in description. Here we added images, videos, background voice, and animation. This will increase interest of students to do more study.
Chapter number 3, Structure of Atom. In this chapter, we'll study prominent scientist, introduction, discovery of electron, discovery of proton, discovery of neutron, concept of atomic number, isotopes, isobars, atomic models, that is Thomson model, Rutherford's atomic model and its limitations, Bohr's model for hydrogen and hydrogen-like atoms, concept of shells and subshells, dual nature of matter and light, de Broglie equation, Heisenberg's uncertainty principle, concept of atomic orbitals, quantum numbers, shapes of orbitals, rules for filling electrons in orbitals, above principle, Pauli's exclusion principle, Hund's rule of maximum multiplicity, electronic configuration of the elements, atomic numbers from 1 to 30, stability of half-filled and completely filled orbitals. Prominent scientists. An English physicist, Sir J. Thomson, was a Nobel Prize winner in the year 1906 for the discovery of electron and the work on conduction of electricity through gases. He also discovered isotopes and invented mass spectrometer. Rutherford, a New Zealand-born chemist, discovered the nucleus of an atom. He was also the Nobel Prize winner of the year 1908. He proposed Rutherford's model of an atom. He named alpha and beta particles to describe two types of radiations emitted by uranium and thorium. In 1919, he became the first person to transmute one element into another and hydrogen into oxygen through nuclear reaction. In 1920, Rutherford proposed existence of a neutral particle, neutron. Niels Bohr was a Danish physicist, did a research on atomic structure and quantum mechanics. He was awarded a Nobel Prize in 1922. His work identified the isotope of uranium-235U was responsible for slow neutron fission. Introduction. Several philosophers believed that matter is composed of extremely small and indivisible particles called atoms. Greek word means indivisible. In 1808, Dalton established a new theory which states that first one, matter consists of indivisible atoms. Second one, the atoms of a particular element have identical properties. The atoms of different elements differ in properties. Third one, when atoms of different elements combine in a fixed ratio of whole numbers, then compounds are formed. Fourth one, in a chemical reaction, rearrangement of atoms take place. The atoms are neither created nor destroyed in a chemical reaction. The atom is conserved. After several experiments at the end of 19th century, it is established that an atom can be subdivided and it contains fundamental particles, electrons, protons and neutrons known as fundamental particles, that is subatomic particles. Chapter number 3, Structure of Atom. In this chapter, we'll study prominent scientist, introduction, discovery of electron, discovery of proton, discovery of neutron, concept of atomic number, isotopes, isobars, atomic models, that is Thomson model, Rutherford's atomic model and its limitations, Bohr's model for hydrogen and hydrogen-like atoms, concept of shells and subshells, dual nature of matter and light, de Broglie equation, Heisenberg's uncertainty principle, concept of atomic orbitals, quantum numbers, shapes of orbitals, rules for filling electrons in orbitals, Aufbau principle, Pauli's exclusion principle, Hund's rule of maximum multiplicity, electronic configuration of the elements, atomic numbers from 1 to 30, stability of half-filled and completely filled orbitals. Prominent scientists. An English physicist, Sir J. Thomson, was a Nobel Prize winner in the year 1906 for the discovery of electron and the work on conduction of electricity through gases. He also discovered isotopes and invented mass spectrometer. Rutherford, a New Zealand-born chemist, discovered the nucleus of an atom. He was also the Nobel Prize winner of the year 1908. He proposed Rutherford's model of an atom. He named alpha and beta particles to describe two types of radiations emitted by uranium and thorium. In 1919, he became the first person to transmute one element into another and converted nitrogen into oxygen through nuclear reaction. In 1920, Rutherford proposed existence of a neutral particle, neutron. Niels Bohr was a Danish physicist, did a research on atomic structure, and Thomson performed several experiments with cathode rays of different energies and strengths of electric field or magnetic fields. He determined the value of E upon m, which is the ratio of charge to mass of particles of cathode rays, as E upon m is equal to 1.758820 into 10 E inverse kg, where m is equal to mass of particle of cathode rays in kilograms. E is equal to charge in coulombs. The particles are negatively charged and therefore the charge on the cathode ray particle is represented as minus E. Sir J. Thomson showed that the value of E upon m for a cathode ray particle was the same irrespective of the nature of gas used in the discharge tube and the nature of material of which the cathode was made. This proved that all the constituent particles of cathode rays are identical. This also proved that these particles are the universal constituents of all the atoms of all matter. Charge on the electron. Scientist R. A. Millikan established a method known as oil drop experiment to determine the charge on the electrons. He found that the charge on the electron is equal to minus 1.6 into 10 raised to minus 19 coulomb. Accepted value of electrical charge is minus 1.6022 into 10 raised to minus 19 coulomb. By knowing charge and E upon ME value, it was possible to determine the mass of electron. ME is equal to E upon M is equal to 1.6022 into 10 raised to minus 19 coulomb divided by 1.75880 into 10 raised to 11 kg inverse, where E is electronic charge and M its mass, which is equal to 9.1094 into 10 raised to minus 31. Mass of electron is found to be 1 upon 1837 the mass of an atom of hydrogen. Discovery of proton. Goldstein carried out electrical discharge experiment in the modified cathode ray tube. He found out that if a perforated disc cathode was used in place of disc cathode and gas pressure was not too low, through the holes of the cathode rays emerged on the opposite of the anode. These rays passed through the holes of the cathode, traveled in the form of a stream on canal and produced a glow on the other end of the discharge tube. Goldstein called them canal rays or anode rays. These rays were found to consist of positively charged particles. Therefore, these rays are also known as positive rays. Properties of canal rays. Canal rays travel in a straight line in the direction opposite to the cathode rays. Second one, canal rays deflect towards the negative electrode in the presence of electric field. This indicates that constituent particles of canal rays are positively charged. Third one, when they fall on the zinc sulfide film, they produce fluorescence. Fourth one, they are affected by a magnetic field. Hence, it is confirmed the presence of positively charged particles. The charge to mass ratio of particles depend on the gas in the discharge tube. And lastly, positively charged particles carry whole number multiple of the fundamental unit of positive electrical charge. Discovery of proton. The lightest positive particle having a unit positive charge could be obtained by using hydrogen gas in the discharge tube. The value of E upon m for the constituent particles of hydrogen gas was found to be minimum. These particles possess minimum mass and a unit positive charge. Determination of mass of these particles revealed that the mass of one particle was 1837 times the mass of an electron. This particle was called proton. When high-speed electrons, that is cathode rays, strike molecule of the gas in the discharge tube, they knock out one or more electrons from it. Thus, a positive ion is formed. M plus E minus gives M plus plus 2 electrons, or M plus n number of electrons gives M plus plus n plus 1 electrons. When electric discharge is passed through the gas under high electric potential, these positive ions pass through the perforated cathode and appear as positive rays. Sir J.J. Thomson showed that, first one, the actual mass of proton is 1.672 into 10 raised to minus 24 gram. The mass of proton is 1.00727 atomic mass unit, which is AMU. Second one, the electrical charge of proton is equal in magnitude but opposite in sign to that of the electron. Thus, proton carries a charge plus 1.60 into 10 raised to minus 19 coulombs, or plus 1 elementary charge unit. Thus, proton is defined as a subatomic particle which has mass of 0.00727 AMU and charge plus 1 elementary charge unit. It has 1 unit mass numbers and 1 unit positive charge. In 1932, Lord Chadwick proved the existence of neutron. Neutron is denoted by 0 and 1. It is neutral in nature, having mass almost equal to mass of proton, as it is a close combination of proton and electron. Heisenberg in 1932 established proton-neutron hypothesis. According to him, an element ZXA has atomic number Z and A the mass number. The nucleus contains Z protons and A minus Z neutrons, and there are Z extranuclear electrons which revolve around the nucleus. Protons and neutrons are present in the nucleus, hence are called nucleons. Therefore, protons, neutrons and electrons are constituents of atoms which are called as subatomic particles. Atomic number. The number of unit positive charges present on the nucleus of an atom of a particular element is called the atomic number of that element. The positive charge which is present on the nucleus of an atom is due to the protons, and each proton has a unit positive charge. Hence, atomic number of an element can also be defined as the number of protons present in the nucleus of an atom of a particular element is called the atomic number of the element. In neutral atom, the number of protons is exactly equal to the number of electrons present in it. Hence, atomic number of an element is equal to the number of electrons present in it. Atomic number is denoted by Z. Therefore, atomic number Z is equal to number of protons is equal to number of electrons. Scientist Moseley proved that there is a relationship between the atomic number Z of an element and the frequency nu of X-rays. Therefore, square root of nu is equal to A into Z minus K, where A and K are constants. Mass number. The mass of the numbers of protons and neutrons present in the nucleus of an atom is called mass number of that atom, and it is represented by A. Thus, mass number A is equal to number of protons plus number of neutrons is equal to total number of nucleons. Electrons which are present in the space around the nucleus have negligible mass. Hence, the entire mass of an atom is concentrated in the nucleus. For a neutral atom, ZXA with mass number A and atomic number Z, number of electrons is equal to Z. Therefore, number of protons is equal to Z. Therefore, number of neutrons is equal to A minus Z. X represents symbol of an atom. Mass number A is written on the top left of the symbol of an element and the atomic number Z towards its left bottom. Isotope. The atoms of an element which have the same number of protons but different number of neutrons are called isotopes of that element. Characteristics of isotopes. First one, the isotopes of an element have same atomic number but different mass numbers. The nuclei of isotopes contain the same number of protons but different number of neutrons. Second one, chemical properties are mainly determined by number of protons in the nucleus and the number of electrons in the atom. The different isotopes of an element exhibit similar properties. isobars. The atoms of different neighboring elements having the same mass number but different atomic numbers are called isobars. Isobars possess different chemical properties. They occupy different positions in the periodic table. They are different atomic species and possess same number. Atomic models. Thomson's model of an atom. Sir J. Thomson in 1898 proposed the first model of an atom known as J. Thomson's model or plum pudding model of an atom. He proposed that an atom has a spherical shape of radius approximately 10 raised to minus 10 meters, in which the positive charge is uniformly distributed. The electrons are embedded uniformly into it to give the most stable electrostatic arrangement. The mass of an atom is assumed to be uniformly distributed over the atom. This model could explain the overall neutrality of the atom but failed to explain origin of atomic spectrum and alpha particle scattering experiment. Discovery of radioactivity. A French physicist, Becquerel, in 1896 proved that atoms are divisible with the help of discovery of radioactivity. The radioactivity is the spontaneous decomposition of atoms of certain unstable elements like radium, uranium, thorium, etc. into atoms of other elements, followed by emission of some smaller charged particles and neutral radiations. When these particles were tested for electrical charge by passing through strong electric field, the behavior was classified into three types. First one, the positively charged particles were found to deflect from the path towards negative electrode. These positively charged particles were named as alpha particles. Second one, the second type of particle ejected from radioactive source was found to deflect towards positive plate. The extent of deflection was much more than the alpha particles. Hence, these particles must be negatively charged and the mass of these particles must be very small as compared to alpha particles. These are named as beta particles. Third one. The third type of radiation was neutral radiation, not deflecting in electric field, was named gamma rays. Rutherford's atomic model. Rutherford in 1909 carried out an alpha particle experiment. He bombarded the beam of alpha particles on a thin gold foil of a thickness 100 nanometer. The gold foil was surrounded by fluorescent screen of zinc sulfide (ZnS). When alpha particles strike the screen, a tiny flash of light was produced at that point. From this experiment, Rutherford observed that first one, most of the alpha particles passed through the gold foil without deflection. Second one, some of the alpha particles were deflected by a large angle greater than 90 degrees. And third one, very few alpha particles were deflected through an angle of 180 degrees. On the basis of these observations, Rutherford suggested his model of an atom. First one, an atom consists of positively charged, massive and dense part at its center called nucleus. Second one, nucleus contains tiny positively charged particles called protons, and almost entire mass of an atom is concentrated at the nucleus. Third one, negatively charged electrons revolve around the nucleus in various circular orbits with high velocity. Fourth one, atom as a whole is electrically neutral. Therefore, the number of protons in the nucleus is always equal to the number of electrons around the nucleus. Fifth one, the electrons and the nucleus are held together by electrostatic force of attraction. Drawbacks of Rutherford's atomic model. First one, according to Rutherford, electrons revolve around the nucleus in different orbits. According to the theory of electrodynamics, a revolving charged body radiates energy continuously. This radiation of energy is possible only at the cost of the electron's kinetic energy. Due to this loss of energy, the radius of the revolving orbit should become smaller and smaller, and electron should come closer to the nucleus and finally fall into the nucleus. But this behavior of the electrons has never been observed. Second one, it should give a continuous spectra, but the observed atomic spectra contains number of lines of different wavelengths. Electromagnetic radiations. Radiant energies like microwave, radio waves, infrared, visible light, ultraviolet X-rays, gamma rays, etc. possess sine wave nature. All radiations travel at equal speed, that is 3 into 10 raised to minus 8 meter per second. Different radiations have different frequencies and different wavelengths. Frequency nu of radiation wave is the number of cycles of wave that pass a given point in a unit time. It is measured in the unit of cycles per second, CPS. In SI unit, it is measured in hertz, denoted by Hz and 1 Hz, that is 1 Hz, is equal to 1 CPS, that is 1 cycles per second. Wavelength lambda is the distance between any two nearest identical points on the wave. It is measured in meters. Smaller unit is micrometer. Mu m is equal to 10 raised to minus 6 meter or nanometer. Nm, that is nanometer, is equal to 10 raised to minus 9 meters. The velocity c, frequency nu, and wavelength lambda of electromagnetic radiation are related by equation nu is equal to c upon lambda. Wave number is the number of waves in centimeter path. It is denoted by nu bar and is measured in the unit centimeter inverse, that is per centimeter. Nu bar is equal to 1 upon lambda. Therefore, nu is equal to C nu bar, or nu bar is equal to nu upon C. Quantum theory of radiation. In 1900, a German physicist, Max Planck, established a theory of black body radiations. He stated that first one, absorption or emission of energy in the form of radiation does not take place continuously. It takes place discontinuously in the form of small packets of energy or quanta or photons. Second one, the quantity of energy E associated with each quantum for a particular radiation is proportional to its frequency nu. Therefore, E is equal to h nu, which is equal to h upon lambda, as nu is equal to 1 upon lambda, where E is equal to energy of a quantum of the radiation. H is equal to Planck's constant having value 6.625 into 10 raised to minus 34 joules per second. Nu is equal to frequency of radiations. Lambda is equal to wavelength of radiation. That is, energy depends upon frequency of radiation. Third one, excited electrons emit energy in any whole number multiple of this unit, and this is transmitted as waves. Bohr's model of an atom. In 1913, Niels Bohr proposed his model of atom on the basis of Rutherford's atomic model and the quantum theory of radiation. Bohr made some assumptions for the stability of an atom. Postulates. First one, electrons revolve around the nucleus in fixed circular orbits called stationary orbits or shells. Second one, as long as electron moves in a given orbit, it does not radiate energy. Hence, the energy of electron remains constant in the same orbit. Third one, each stationary orbit corresponds to a certain fixed energy level. These energy levels are numbered as 1, 2, 3, 4 or KLMN, etc. from the nucleus of an atom. Fourth one, energy goes on increasing as the energy levels go away from the nucleus, that is K less than L less than M less than N. Fifth one, energy transition takes place when an electron jumps from one energy level to another. If it jumps from a lower energy level E1 to the higher energy level E2, the difference of energy E2 minus E1 is absorbed in the form of quantum, while energy is given out when electron jumps from higher energy level to the lower one. Sixth one, only those orbits are possible for which the angular momentum is an integral multiple of h upon 2 pi, that is mvr is equal to n into h upon 2 pi, where m is equal to mass of electron, v is velocity of electron, r is radius of orbit, n is equal to 1, 2, 3 number of shells, h is equal to Planck's constant, which is equal to 6.626 into 10 raised to minus 34 joules seconds. Drawbacks of Bohr's atomic model. First one, Bohr's theory was able to explain the formation of spectrum of simple atoms like hydrogen and helium. It failed to account for the spectrum of the atoms that have several electrons. Second one, when the atomic source of radiation is placed in a magnetic field, a line in an emission spectrum splits up into a number of closely packed lines. This effect is called as Zeeman effect, and Bohr's theory could not explain this effect. Third one, when the atomic source of radiation is placed in an electrical field, a line in an emission spectrum splits into a number of closely packed lines. This effect is called as Stark effect, and Bohr's theory could not explain this effect. Lastly, it failed to take into consideration the dual nature, that is, as a wave and as a particle nature of subatomic particles. Concepts of shell and subshells. According to Bohr's atomic model, electrons revolve around the nucleus in stationary energy levels called orbits. These orbits are also called as principal energy levels or shells. These energy levels are represented by principal quantum number n, which assumes only integral values 1, 2, 3, etc. and are represented by letters K, L, M, N, etc. Subshells. Each shell is divided into subshells. They are designated as SPDF. Each subshell in a shell is assigned an azimuthal quantum number L. For a given value to n, L can have n values ranging from 0 to n minus 1. Magnetic quantum number M describes number of orientations of an electron present in a subshell, that is, number of orbitals in a given subshell in presence of magnetic field. Total number of value of M is equal to 2L plus 1. For each value of L, M has integral values from minus L to 0 to plus 1. Spin quantum number S gives the direction of the spin of the electron in an atom, which may be clockwise or anticlockwise. Clockwise spin is represented by upwards arrow having value plus half. Anticlockwise spin is represented by downwards arrow having value minus half. Wave motion and its characteristics. A wave is a periodic disturbance in space, and wave motion represents the propagation of this periodic disturbance which carries energy. Wavelength lambda. The distance between two consecutive crests or two consecutive troughs is called wavelength, represented by lambda. It is measured in angstrom units A or nanometer nm. Frequency nu. The number of waves passing through reference point in 1 second is called the frequency, represented by nu. It is expressed in the units cycles per second CPS or Hertz Hz. Velocity c. The distance travelled by a wave in 1 second is velocity of the wave. It is denoted by letter c. It is also the product of the frequency and wavelength. Hence, c is equal to nu into lambda. Wave number nu bar. The number of waves which can be accommodated in 1 centimeter path of radiation is called as nu bar. It is expressed in the unit centimeter per 1. Wave number of a wave is equal to the reciprocal of its wavelength in centimeters. Nu bar is equal to 1 upon lambda. Amplitude A. The height of a crest or the depth of trough from the line of propagation of the wave is called amplitude, represented by A. The amplitude of the wave determines the intensity of the radius electromagnetic spectrum. The arrangement of different types of electromagnetic radiations in order of increasing wavelength is called electromagnetic spectrum. Photoelectric effect. When a beam of light of sufficient energy is made to fall on the surface of a metal electrons are ejected. This phenomenon is known as photoelectric effect, and the electrons thus ejected are known as photoelectrons. De Broglie's equation. An electron behaves as both as a particle and as a wave. De Broglie suggested that all material objects show a dual nature. Every object which possesses a mass and velocity behaves both as a particle and as a wave. According to de Broglie, the wavelength lambda of a particle of mass m moving with a velocity nu is given by lambda is equal to h upon mv, where h is Planck's constant. The quantity mv represents momentum of the particles. The above equation can also be expressed as lambda is equal to h upon p, where p stands for momentum of a particle. From the above equation, it is clear that the wavelength of a moving particle is inversely proportional to its mass. Heisenberg's uncertainty principle. In 1927, Heisenberg, a German physicist, proposed uncertainty principle. It can be stated as it is not possible to determine simultaneously the position and momentum of a moving microscopic particle, that is electron, with absolute accuracy. Orbital. The three-dimensional region in space around a nucleus where the probability of finding an electron is maximum is known as orbital. There are four types of orbitals. S orbital having spherical shape. Second one. P orbital has dumbbell shape. Third one. D orbital is four-lobe planar structure. F orbital is complicated three-dimensional structure. S orbital. S orbital is spherical in shape and non-directional. It has only one orientation in space for each main energy level. In an atom, there is one s orbital. The size and energy of s orbital increase as we go to higher energy levels of an atom, that is 1s less than 2s less than 3s less than 4s. P orbital. For p orbital, m is equal to -1, 0, +1. Therefore, electron in p orbital has three orientations in space. These are present along the XYZ axis. They are designated as Px, Py, Pz. These three p orbitals are right angles to each other. Each p orbital is dumbbell shaped with two lobes projecting opposite side of the nucleus. In each p orbital, the electron has a definite energy. As the electron moves away from the nucleus, its energy also go on increasing. Quantum numbers. It is a set of 4 numbers with the help of which the complete information about all electrons in an atom, that is, information regarding location, energy, direction of spin. The 4 quantum numbers are principal quantum number denoted by N, azimuthal quantum number denoted by L, magnetic quantum number denoted by M, and lastly spin quantum number denoted by S. The position of an electron in the main shell N, subshell L, orbital M, and its spin S can be completely described by its 4 quantum numbers. Rules for filling electrons in orbital. First one, electronic configuration. The arrangement of electrons around the nucleus in an atom is called electronic configuration. Electronic configuration of an atom is the distribution of electrons in shells and subshells and orbitals around the nucleus of an atom. It is written as NL raised to X, where N is equal to shell number, L is equal to subshell, X is equal to number of electrons in subshell. Electronic configuration is mainly explained by following rules. First one, outbow principle. Outbow means building up. Second one, Pauli's exclusion principle. And third one, Hund's rule of maximum multiplicity. First one, outbow principle. Electrons are added progressively starting with the orbital of the lowest energy to the various other orbitals in the order of their increasing energy. Hence, orbitals of lower energy are filled up first and then higher energy orbitals are filled up progressively by the electrons. The sequence in which the orbitals are filled is shown below. Order of filling of atomic orbitals by the electron, it is given, please observe it. The order in which the energies of the orbitals increase and hence the order in which the orbitals are filled is as follows. 1s less than 2s less than 2p less than 3s less than 3p less than 4s less than 4p less than 3d less than 4p less than 5s less than 5d less than 5p less than 6s less than 4f less than 5d less than 6p less than 7s and less than 5f. Second one, Pauli's exclusion principle. On the basis of spectroscopic evidences and from the study of quantum numbers, Pauli in 1925 established a principle known as Pauli's exclusion principle, which gives the number of electrons that can occupy any orbital in that it is impossible for any two electrons in the same atom to have all the four quantum numbers identical, or two electrons in an atom cannot have the same set of all the four quantum numbers. An orbital can accommodate maximum of two electrons and they must possess opposite spins. Third one, Hund's rule of maximum multiplicity. This rule deals with the filling of electrons in equal energy orbitals of same subshell P or D or F. It states that when several orbitals of the same energy, that is degenerate orbitals, are available, the electrons first fill all the orbitals with parallel spin singly before pairing in any one orbital. For example, 4 electrons in the p orbital are filled accordingly. Please observe it. Anomalous behavior of chromium Cr and copper Cu. First we will consider anomalous behavior of chromium, that is Cr. Atomic number of chromium is 24. Expected electronic configuration 1s2, 2s2, 2p6, 3s2, 3p6, 4s2, 3d4. Empty, half-filled and the completely filled orbitals are more stable than those of incompletely filled orbitals. When one more electron is added to 3d4, the outermost orbital acquires the configuration of half-filled orbital, that is 3d5, and becomes more stable. As the energy difference between 3d and 4s orbitals is very small, one electron from 4s orbital jumps to 3d orbital to make it half-filled and thus attains stability. Thus, electronic configuration of chromium becomes 1s2, 2s2, 2p6, 3s2, 3p6, 4s1 and 3d5. Anomalous behavior of copper Cu. Atomic number of copper is 29. Its expected electronic configuration is 1s2, 2s2, 2p6, 3s2, 3p6, 4s2, 3d9. If one more electron is added to 3d9, it acquires the configuration of a completely filled orbital 3d10 and becomes more stable. The energy difference between 3d and 4s orbital is very small. An electron from 4s orbital jumps to 3d orbital easily, means 3d orbital attracts one electron from 4s orbital and 3d orbital becomes completely filled and attains more stability because empty, half-filled and completely filled orbitals are more stable. Thus, the electronic configuration of copper becomes 1s2, 2s2, 2p6, 3s2, 4s1, 3d10. Now please observe the electronic configurations of first 30 elements.
Now friends, the Chemistry 1 chapter is complete and we are going to Chemistry 2. In Chemistry 2, study is given. In study, all topics is Chemistry 2. In topic, hydrogen we are studying. In hydrogen, only introduction is given. In introduction, not only textual information provided, but also we add images, background work, videos, and animation. This will help to understand each topic properly and increase your knowledge.
Hydrogen. Introduction. Position of hydrogen in periodic table. Occurrence of hydrogen, that is dihydrogen. Isotopes of hydrogen. Preparation of dihydrogen. Properties of dihydrogen. Uses of dihydrogen. Hydrides, that is ionic, covalent and interstitial. Water, physical and chemical properties of water. Heavy water. Hydrogen peroxide, that is preparation, properties and structure. Hydrogen as a fuel. Introduction. Hydrogen, the first element in the periodic table. Electronic configuration is 1s1. Contains only one proton and one electron. Exists as a diatomic molecule, hence is called as dihydrogen. Most abundant element in the universe. Combines with other elements by losing, gaining and sharing of electrons. The electronic configuration of hydrogen 1s1 is similar to the outer electronic configuration ns1 of alkali matrix. Alkali matrices are placed in the first group of the periodic table. Hence, hydrogen also can be a member of the first group. Like halogens Ns2Np5, hydrogen also contains one electron less than the corresponding noble gas configuration helium 1s2, and accordingly it can also be a member of group 17. Thus, hydrogen has resemblance to alkali matrix, which can lose one electron to form unipositive ions, as well as with halogens, which gain electron to form uninegative ions. Hydrogen forms halides, oxides and sulfides like alkali matrix, and has very high ionization enthalpy and does not possess metallic character under normal conditions. On the other hand, alkali matrix exhibits metallic character. Considering ionization enthalpy, hydrogen resembles more with halogens. Hydrogen also forms diatomic molecules such as hydrides and large number of covalent compounds like halogens. But hydrogen is very less reactive as compared to halogens. Thus, hydrogen resembles both the alkali matrix and the halogens. Dihydrogen is the most abundant element in the universe. 70% of the total mass of the universe and is the main element in the solar system. In the combined form, it is found in the Earth's crust, oceans, plants and animal tissues, carbohydrates, proteins, hydrides, hydrocarbons and many other compounds. Hydrogen has three isotopes: protium 1H1, deuterium 1H2 or D, and tritium 1H3 or T. Tritium is radioactive and is a beta particle emitter. Its half-life is 12.33 years. These three isotopes have 0, 1 and 2 neutrons in the nucleus respectively. Their physical properties are different due to their different masses, but chemical properties are similar due to their identical electronic configuration. This effect is known as isotopic effect. The following table shows atomic and physical properties of hydrogen, deuterium and tritium. Property: hydrogen, deuterium and tritium. Active abundance in percentages: 99.965, 0.0156 and 10 raised to minus 15. Relative atomic mass in grams per mole: 1.008, 2.014, 3.016. Melting point in Kelvin and boiling point in Kelvin. Melting point is 13.96, 18.73 and of tritium is 20.62. Boiling point in Kelvin is 20.39, 23.67 is of deuterium and 25.0 is of tritium. Density in grams per liter: 0.09, 0.18 and 0.27. Enthalpy of fusion in kilojoules per mole: 0.117, 0.197. Enthalpy of vaporization in kilojoules per mole: 0.904, 1.226. Enthalpy of bond dissociation in kilojoules per mole at 298.3 Kelvin. 435.88 is of hydrogen, 443.35 is of deuterium. Intermolecular distance in picometers: 74.14, 74.14 of deuterium. Ionization enthalpy in kilojoules per mole: 1312. Electron gain enthalpy in kilojoules per mole: minus 73 is of hydrogen. Covalent radius in picometers: 37 is of hydrogen. Ionic radius, that is of hydrogen minus, in picometer: 208. Laboratory preparation of dihydrogen, that is H2. Dihydrogen is prepared by using metals and metal hydrides. First one, zinc plus twice NaOH gives Na2ZnO2 plus hydrogen gas evolves. Second method, zinc plus twice HCl gives ZnCl2 that is zinc chloride plus hydrogen gas evolves. Third method is magnesium plus dilute H2SO4 gives magnesium sulfate and hydrogen gas evolves. Fourth method, sodium hydride, that is NaH plus H2O gives sodium hydroxide NaOH, and in the reaction hydrogen gas evolves. Fifth method is twice of aluminium plus twice of potassium hydroxide KOH plus twice of H2O gives twice of KaLO2 plus 3 hydrogen molecules. Commercial production of dihydrogen. First one, by electrolysis of acidified water using platinum electrodes, and second method, by electrolysis of warm aqueous solution of barium hydroxide between nickel electrodes. Third method is from water gas, that is Bosch process. Fourth one is by electrolysis of brine solution in the manufacture of sodium hydroxide and chlorine. Fifth one, reaction of steam on hydrocarbons or coke at high temperatures. Sixth method is from steam, that is Lenz process. Properties of dihydrogen. Physical properties. It's odorless, colorless, tasteless, combustible gas, lighter than air and insoluble in water. Chemical properties. Hydrogen-hydrogen bond dissociation enthalpy is 435.88 kilojoules per mole at 298 Kelvin is very high, hence it is relatively inert at room temperature and is not very active. Thus, atomic hydrogen is produced at high temperature in an electric arc or under ultraviolet radiations. Its electronic configuration is 1s1, means its orbital is incomplete, hence it combines with almost all elements. Reactions of hydrogen take place due to loss of electron to form H plus ion. Secondly, gain of electron to form hydrogen minus ion. And lastly, sharing of electrons to form single covalent bond. Uses of dihydrogen. It is used in the manufacture of vanaspati fat by hydrogenation of polyunsaturated vegetable oils like soybean, cottonseed, etc. In the synthesis of ammonia, which is used in the manufacture of nitric acid and nitrogenous fertilizers. In the manufacture of methanol. For the preparation of hydrogen chloride. For the manufacture of metal hydrides, ionic hydrides, covalent hydrides and interstitial hydrides. Used as rocket fuel in space research, liquid hydrogen plus liquid oxygen. Used in fuel cell for generating electrical energy. It does not produce pollution and releases greater energy per unit mass of fuel in comparison to gasoline and other fuels. In the reduction of heavy metal oxides to metals in metallurgical process. Atomic hydrogen and oxy-hydrogen torches are used for cutting and welding purposes to generate the temperature of 4000 Kelvin. Hydrides. Under specific conditions, dihydrogen combines with almost all elements to form binary compounds called hydrides. For example, lithium hydride, LiH, magnesium hydride, magnesium MgH2, B2H6, that is boron hexahydride, etc. These are classified as: first one, ionic or saline or salt-like hydrides. Second one, covalent or molecular hydrides. And lastly, metallic or non-stoichiometric or interstitial hydrides. Water. Water is the most precious gift to mankind, which is the most essential commodity for the existence of life. The distribution of water over the Earth's surface is not uniform. Physical properties of water. Water is colorless, tasteless liquid. Properties and water. First one, molecular mass in grams per mole, that is 18.0151. Enthalpy of formation in kilojoule per mole is minus 285.9. Enthalpy of fusion in kilojoules per mole 6.01. Enthalpy of vaporization is 40.66. Melting point in Kelvin 273. Boiling point in Kelvin 373. Temperature of maximum density in grams per centimeter cube is 276.98. Density at 298 Kelvin in grams per centimeter cube is 1.00. Dielectric constant is 78.39. Electrical conductivity at 293 Kelvin per ohm per centimeter 5.7 into 10 raised to minus 8. Viscosity in centipoise 0.8903. Water molecules possess extensive hydrogen bonding. Structure of water. In the gas phase, water is a bent molecule and highly polar molecule. The crystalline form of water, that is ice, at atmospheric pressure, ice crystallizes in the hexagonal form. At very low temperature, it condenses to cubic form. The density of ice is less than that of water. Hence, ice floats on water. Ice has a highly ordered three-dimensional hydrogen bonded structure. Hydrogen bonding gives ice an opaque type structure with wide holes. These holes can hold some other molecules of appropriate size interstitially. Now we'll observe the structure of ice. Chemical properties of water. First one, amphoteric character. Water acts as both as an acid and as a base. Such behavior is known as amphoteric character. Second one, redox reaction involving water. Water can be easily reduced to dihydrogen by highly electropositive matrix. Third one, hydrolysis reactions. Water has high dielectric constant, hence it has a very strong solvating tendency. Fourth one, formation of hydrates. From aqueous solution, many salts can be crystallized as hydrated salts. Such association of water is of different types. First one, coordinated water. For example, CrH2O6 plus 3 thrice Cl minus 1. Second one, interstitial water. BaCl2 twice H2O. Third one, hydrogen bonded water. CuH2O4 plus 2 SO4 minus 2 H2O in CuSO4 5 H2O. Heavy water. Heavy water is deuterium oxide D2O. Hydrogen has 3 isotopes. The most common 1H1 has no neutron. Its nucleus is only a proton. The isotope 1H2 is called deuterium D with a nucleus that consists of 1 proton and 1 neutron and has about twice the mass of an ordinary hydrogen atom. It is 1.998 times heavier. On the combination of oxygen, it forms heavy water. Physical properties of D2O. Property and D2O. Molecular mass in grams per mole is 20.0276. Melting point in Kelvin 276.8. Boiling point in Kelvin 374.4. Enthalpy of formation in kilojoules per mole minus 294.4. Enthalpy of vaporization at 373 Kelvin in kilojoule per mole is 41.61. Temperature of maximum density in grams per centimeter cube is 284.2. Density at 298 Kelvin in grams per centimeter cube is 1.1059. Viscosity in centipoise 1.107. Dielectric constant 78.06. Heavy water is used in nuclear reactors as moderator for neutrons. The isotope 1H3 is called tritium T. The tritium nucleus has 1 proton and 2 neutrons, and so it is about 3 times as heavy as hydrogen. When it is combined with oxygen, it forms super heavy water T2O. It freezes at 9 degrees Celsius, boils at 104 degrees Celsius, and its density at 20 degrees Celsius is 1.88 grams per centimeter cube. Oxygen has three isotopic forms: O16, O17 and O18. Thus, there are 18 different kinds of water, and it is very difficult chemically to separate out all these isotopic varieties from the sources. The following are the isotopic varieties of water: 16H2O, 16D2O, 16T2O, 16HDO, 16HTO, 16D2O, 18, 20, 22, 19, 20, 21. H217O, D217O, T217O, HD17O, HT17O, DT17O, 19, 21, 23, 20, 21, 22. H218O, D218O, T218O, HD18O, HT18O, DT18O, 20, 22, 24, 21, 22, 23. From the above table, it is observed that the lightest water is H218O and heaviest is T218O, whose molecular weights are 18 and 24 respectively. Among different kinds of water, protonium water or ordinary water is vital for life process. Other isotopic forms of water are important in reaction kinetics. Hydrogen peroxide H2O2, that is oxygenated water. Preparation of hydrogen peroxide. First one, laboratory method. Using acidified barium peroxide, that is BaO2. Second one, Merck's method, by action of dilute acids on sodium peroxide, Na2O2. Third one, by bubbling carbon dioxide through a paste of BaO2. Fourth one, by the action of phosphoric acid on BaO2. Fifth one, manufacture of H2O2 by electrolysis of 50% sulfuric acid. Sixth one, industrially it is prepared by auto-oxidation of 2-ethyl anthraquinol. Physical properties of H2O2. Pure H2O2, that is hydrogen peroxide, is very pale blue liquid. Melting point in Kelvin is 272.4. Vapor pressure at 298 Kelvin in millimeter of mercury, that is 19. Density solid at 268.5 Kelvin per gram per centimeter, 164. Density liquid at 298 Kelvin per gram per centimeter is 1.44. Viscosity at 290 Kelvin in centipoise, 70.7. Dielectric constant at 298 Kelvin per Newton per meter square 1.25. Electrical conductivity at 298 Kelvin per meter per centimeter is 5.1 into 10 raised to minus 4. Hydrogen peroxide H2O2 is miscible with water and forms a hydrate H2O2 H2O. Melting point is 221 Kelvin. A 30% solution of hydrogen peroxide is marked as 100 volume hydrogen peroxide. 1 milliliter of 30% H2O2 solution will give 100 volume of oxygen at STP. Commercially, it is marketed as 10 volume, which means it contains 3% H2O2. Structure of hydrogen peroxide. It is a non-planar open book structure. Hydrogen oxygen, that is OH single bond length, is 95 picometer. Oxygen bond length is 147.5 picometer. HOH bond angle is 94.8 degrees. Angle between the planes containing HOO is 111.5 degrees in gaseous state. The bond length and bond angles are different in liquid and solid phases due to hydrogen bonding. In crystalline state, the bond angle between two planes reduces to 90.2 degrees. Hydrogen peroxide is very important in industries. Chemical properties of H2O2. First one, it decomposes on heating in presence of finely divided metals such as Ca, that is calcium, Fe, that is iron, copper, Cu, Au, gold, Ag, silver, Pt, platinum, MnO2, manganese dioxide, carbon, dust, light, etc. It is diamagnetic and exhibits dipole moment 2.1 d. It acts as a weak acid and forms salts such as hydroperoxides and peroxides. It neutralizes alkali. Second one, it acts both as an oxidizing and reducing agents in both acidic and alkaline media. Third one, it reduces Cl2 to HCl. It is called as anti-chlor. It also acts as a bleaching agent. It forms glycols with alkenes. Dihydrogen as a fuel. Dihydrogen releases large amount of energy, nearly 3 times more than petrol. As compared to petrol, dihydrogen produces less pollution.
Then, hello students. Now the complete hydrogen introduction part and chemistry second chapter is complete. Now we are going to next chapter is physics 1. Physics 1 study is given. All chapter is given. After that, for example, in this topic, introduction, this is topic force. In this topic, introduction part is given. In introduction, not only textual information is given, but also images, background voice, videos, and animations are given, which will help to improve your knowledge and understand topic more easily.
Chapter 4, Force. Newton's law of motion. Newton's first law states that everybody continues to be in a state of rest or of uniform motion along a straight line unless it is acted upon by an external unbalanced force. Newton's second law states that the rate of change of linear momentum of a body is directly proportional to the external unbalanced force applied on the body and takes place in the direction of force. Newton's third law states that to every action or force there is always an equal and opposite reaction. According to Newton's second law, force is equal to P2 minus P1 upon T1. Impulse of the force or impulse F into T is equal to P2 minus P1. This shows that impulse is equal to the change in linear momentum of the body. Impulse is a vector quantity. Its direction is same as that of the force. The SI unit of impulse is kilogram meter per second or NS. That is same as that of linear momentum. Dimensions of the impulse are L raised to 1, M raised to 1, T raised to minus 1. A force which is produced due to interaction between the objects is called real force. In order to apply Newton's laws to explain some situation, we need to introduce an extra force term in the equation of motion. This added term, which does not have a real origin, is called pseudo force. The existence of the force of attraction between a body and the earth is universal. This force is called gravitational force of attraction. Newton's law of gravitation. Every particle of matter attracts every other particle with a force whose magnitude is directly proportional to the product of their masses, inversely proportional to the square of distance between them, and independent of the medium. This force is directed along line joining the centers of the two particles. Let m1 and m2 be masses of two particles separated by a distance r. The magnitude of force of attraction, called as gravitational force of attraction, is given by F is equal to G into m1 m2 upon r square, where G is constant, called the universal constant of gravitation. G is equal to 6.67 multiplied by 10 raised to minus 11 Newton meter square per kilogram square. Characteristics. This force is always attractive and applicable for microscopic and macroscopic bodies. Its range is unlimited and it is very weak as compared to other real forces. It obeys inverse square law of distance. It does not depend on intervening medium. Electromagnetic forces. Forces between electric charges in relative motion are called electromagnetic forces. The force holding the atoms and molecules of a body together is called electromagnetic force. Characteristics. These forces can be attractive or repulsive. Electromagnetic forces are stronger than that of gravitational forces. They obey inverse square law of distance. They depend on intervening medium. They are long-range forces. Nuclear forces. Nuclear forces are of two types. The force of strong interaction, which binds protons and neutrons in the nucleus of an atom. The force of weak interaction, which results in the decay of many unstable nuclei. Characteristics of strong nuclear force. Nuclear force is attractive and has short range, nearly 10 raised to minus 14 meters. It is the strongest force in nature. It does not obey inverse square law of distance. Nuclear forces are charge independent. Force between Pp, Nn and Pn have some magnitude, that is, not affected by nature of charge. Characteristics of weak nuclear force. It acts between any two elementary particles. It is much weaker than the strong nuclear force or electromagnetic force. It is stronger than the gravitational force. Its range is very short, of the order of size of the nucleus. Law of conservation.
Now friends, we are to the introduction of force. After that, complete the topic with physics 1, and after we will go into next topic is physics 2. The study is given in physics 2. All chapter is given. In this chapter, for example, we will see the electronic chapter. Electronics. In this chapter, introduction is given. In introduction, not only information is given. In introduction, not only information is given, but also images, diagrams, volumes, videos, and animation are added, which will help to understand this topic very easily.
Electrostatic. Introduction. The study of electric charges at rest is known as electrostatics. It deals with forces, fields. When two objects are rubbed with each other, frictional electricity is developed. In this chapter, we will learn about charges, Coulomb's law, superposition principle, distribution of charges, electric field intensity, potential dipole, equipotential surfaces, etc. Explanation. Coulomb's law. The electrostatic force of attraction or repulsion between 2-point electric charges at rest is directly proportional to product of magnitude of two charges and inversely proportional to square of distance between them. F is equal to 1 upon 4 pi epsilon O multiplied by Q1 Q2 upon R square. In vector form, vector F21 is equal to 1 upon 4 pi epsilon O K multiplied by Q1 Q2 upon R square multiplied by R12. Limitation. It is applicable to stationary point charges and not for extended bodies. It can be applied for charged extended objects, provided that distance between them is sufficiently large. Definition of Coulomb. Coulomb is defined as the magnitude of that charge if placed 1 meter apart from a charge of same magnitude will repel the other charge with a force of 9 multiplied by 10 raised to 9 Newton. Dielectric constant. The ratio of permittivity of medium to permittivity of vacuum is called dielectric constant. K is equal to epsilon upon epsilon O. Superposition principle. If there are n number of charges interacting, the total force on a given charge is vector sum of individual forces exerted on given charge by all other charges. Vector F1 is equal to vector F12 plus vector F13 plus vector F14 up to plus vector F1n. Linear charge density. Charge per unit length of conductor is called linear charge density. Lambda is equal to Q upon L. Surface charge density. Charge per unit area is called as surface charge density. Sigma is equal to Q upon A. Volume charge density. Charge per unit volume is called as volume charge density. Rho is equal to Q upon V. Electric field intensity due to a point charge. The electric field intensity due to point charge at any point in the electric field is defined as electrostatic force exerted by it on a unit positive charge placed at that point. E is equal to F upon QO unit N upon C. Derivation. Consider a point charge placed at point O. Consider another point A at distance R. The positive test charge QO is placed at point A. The force experienced by it is vector F is equal to 1 upon 4 pi epsilon OK multiplied by QO upon R square multiplied by RO. By definition of electric field intensity, vector E is equal to vector F upon 90 is equal to 1 upon 4 pi epsilon O K multiplied by Q upon R square multiplied by RO. The magnitude of electric field intensity is given by E is equal to 1 upon 4 pi epsilon O K multiplied by Q upon R square. Electric lines of force. The path along which the unit positive charge moves is called as line of force. Properties. 1. The lines of force start from a positive charge and end on negative charge. The lines of force do not intersect each other. The lines of force do not pass through conductor. The lines of force have tendency to contract along their length. Lines of force exert lateral pressure on each other. Electric lines of force are crowded in a region where electric intensity is large. Electric dipole and electric dipole moment. Two equal and opposite electric point charges separated by finite distance form an electric dipole. Electric dipole moment. The product of magnitude of one of the charge and distance between the two charges forms electric dipole is called as electric dipole moment. P is equal to Q multiplied by 2L.
Then came clear to the introduction. After that, the physics basic chapter is closed and we are going to mathematics. In this subject, study is given. We study all topics are given. For example, we are trigonometric functions of compound angles. In this topic, introduction is given. In this, we are not only providing textual information, but also we are adding images, voice, videos, and animations, which will increase your interest to doing more study and improve your knowledge.
Trigonometric functions of compound angles. An angle formed by the algebraic sum of two or more angles is called a compound angle. For given angles A and B, angles of the form A plus B and A minus B are called the compound angles. Expressions for trigonometric functions of A plus B and A minus B in terms of the trigonometric functions of A and B are called as addition and subtraction formulae. 1. Cos of A minus B is equal to cos A cos B plus sin A sin B. 2. Cos of A plus B is equal to cos A cos B minus sin A sin B. 3. Sin of A plus B is equal to sin A cos B plus cos A sin B. 4. Sin of A minus B is equal to sin A cos B minus cos A sin B. 5. Tan of A plus B is equal to tan A plus tan B divided by 1 minus tan A tan B. 6. Tan of A minus B is equal to tan A minus tan B divided by 1 plus tan A tan B. If the sum or difference of the measures of two angles is either 0 or integral multiple of 90 degrees, that is n pi by 2, where n belongs to set of integers, then these angles are said to be allied angles. Thus, if theta is the measure of a given angle, then the angles minus theta, pi by 2 minus theta, pi by 2 plus theta, pi minus theta, pi plus theta, 3 pi by 2 minus theta, 3 pi by 2 plus theta, 2 pi minus theta are its allied angles. The trigonometric functions of allied angles of theta with respect to trigonometric functions of theta are as shown below. Allied angles minus theta, pi by 2 minus theta, pi by 2 plus theta, pi minus theta, pi plus theta, 3 pi by 2 minus theta, 3 pi by 2 plus theta, 2 pi minus theta. Trigonometric functions. 1. Sine. Sine minus theta is minus sine theta. Sine pi by 2 minus theta is cos theta. Sine of pi by 2 plus theta is cos theta. Sine of pi minus theta is sin theta. Sine of pi plus theta is minus sin theta. Sine of 3 pi by 2 minus theta is minus cos theta. Sine of 3 pi by 2 plus theta is minus cos theta. Sine of 2 pi minus theta is minus sin theta. 2. Cos. Cos of minus theta is cos theta. Cos of pi by 2 minus theta is sin theta. Cos of pi by 2 plus theta is minus sin theta. Cos pi minus theta is minus cos theta. Cos of pi plus theta is minus cos theta. Cos of 3 pi by 2 minus theta is minus sin theta. Cos 3 pi by 2 plus theta is sin theta. Cos of 2 pi minus theta is cos theta. 3. Tan. Tan of minus theta is minus tan theta. Tan pi by 2 minus theta is cot theta. Tan pi by 2 plus theta is minus cot theta. Tan pi minus theta is minus tan theta. Tan of pi plus theta is tan theta. Tan of 3 pi by 2 minus theta is cot theta. Tan of 3 pi by 2 plus theta is minus cot theta. Tan 2 pi minus theta is minus tan theta. Angles of the form 2 theta, 3 theta, 4 theta, etc. are integral multiples of theta. These angles are called multiple angles. Further, angles of the form theta by 2, 3 theta by 2, etc. are called submultiples of theta. A. Trigonometric functions of 2 theta. Result 1. Sin 2 theta is 2 sin theta into cos theta. Cos 2 theta is equal to cos square theta minus sin square theta, which is equal to 1 minus 2 sin square theta, which is equal to 2 cos square theta minus 1. Tan 2 theta is 2 tan theta upon 1 minus tan square of theta. Result 2. Sin 2 theta is equal to 2 tan theta upon 1 plus tan square theta. Cos 2 theta is equal to 1 minus tan square theta upon 1 plus tan.
After finish mathematics 1, we will go into mathematics 2. Study is given in mathematics 2. All the chapters is given. For example, we see a topic Elaboration. In Elaboration, introduction is given. In introduction, we are provided not only textual information, but also we are given images, stories, figures, and animation, which will help to improve your knowledge. Students can do more study very easily.
Logarithm. If a raised to x is equal to y, then x is equal to log y to base a. We read log of y to the base a. For example, 3 raised to 2 is equal to 9. Therefore, 2 is equal to log 9 to the base 3. Second, 4 raised to 0 equal to 1. Therefore, 0 is equal to log 1 to base 4. Third, 9 raised to minus 2 is equal to 1 upon 81. Therefore, minus 2 is equal to log 1 upon 81 to the base 9. Fourth, 25 raised to 1 half is equal to 5. Therefore, 1 half is equal to log 5 to the base 25. Through the above problems, we observe that when x is equal to log of y to base a or a raised to x is equal to y, we have 1. Y greater than 0. Second, a greater than 0 and a not equal to 1. Third, x is any real number. We express the following in exponential form. 1. Log 128 to base 2 is equal to 7. Therefore, 2 raised to 7 is equal to 128. Second, log 16 to base 8 is equal to 4 by 3. Therefore, 8 raised to 4 by 3 is equal to 16. Third, log 0.01 to the base 10 is equal to minus 2. Therefore, 10 raised to minus 2 is 0.01. 4. Log 64 to base root 2 is equal to 12. Therefore, root 2 raised to 12 is equal to 64. 5. Log cube root 5 to base 5 is 1 upon 3. Therefore, 5 raised to 1 upon 3 is equal to cube root of 5. Problems. Evaluate. 1. Log 8 to base 1 half. Let log 8 to base 1 half equal to x. Then 1 half raised to x is equal to 8. Therefore, x is equal to minus 3. Second, log 625 to the base 5. Let log 625 to base 5 be equal to x. Therefore, 5 raised to x will be equal to 625 and therefore x has to be 4.
After that, then mathematics 2 subject is closed. Friends, if you have any requirement of taking the lead of yours, then follow the following information. Our company name, Confer Systems. Our office is office address, office number 6, second floor, Krupaprasad, near Sane Guruji, Smarak, Dandekar Bridge, Singhgad Road, Pune 30. Telephone number 9420694165. Contact for sales 8411890302. 8411900302. 8411910302. 8411920302. 8411940303. 8411950303. Contact for support 84110707 8411090505. If you want to extra of more information, then go to our website www.confersystems.com or www.eguplus.co.in.